Spanish and I speak French. We all speak the same. Yes.
Wouldn't it be a UG since,
Oh, yeah. So sometimes you'll see the code is in DNA language, but I think in the one that you saw, it's RNA language. So yes, so you'll see, sometimes you'll see AUG for methionine. Just depends on how, if we want to read it as DNA or RNA. So, yeah.
Could you go over the biotechnology part of your final study?
The biotechnology part of
your final study?
Oh, yes, I will. So, you know what, were there any other questions on transcription, translation? I'm trying to think about what other details you need to know here. You know where it's all happening. Oh, I know. The one thing that you want to make sure you know is the enzyme that does this. So this here is RNA polymerase. The enzyme that makes RNA, the polymer of RNA, by reading DNA. Now down here, we're really not talking about enzymes, right? Down here, we're talking about tRNA and ribosome. Yes?
Processing also happens.
Yes, processing happens in the nucleus. Yes.
the RNA polymerase is the enzyme that does transcription?
Does transcription, yeah.
Yes.
Things like the E site, the P site, the A site, all that within the ribosome, I don't ask about that.
Yeah. RNA polymerase.
Other questions on transcription translation section? Yes? What is it again?
I'm just checking.
Oh.
Signaling. The P53.
Oh, okay, so let's do a little biotech first, and then I'll come back, and I'll talk about P53 for you. Because it falls into several sections with cancer as well. All right, so can I erase all this? Oh, I want to remind you guys that I do have those lecturettes under your material, like looking each unit's material. And it's just me writing on a PowerPoint slide and talking. So you can walk through some concepts, maybe like aerobic cellular respiration or recombinant DNA technology. All right, so in biotech chapter, a couple things that we want to know, right? You want to understand recombinant DNA technology. You want to understand the Human Genome Project. Okay, I'll talk about each of these. You're going to want to understand what gene therapy is, what CAR T is. That's really in the cancer chapter, but it's here. So CRISPR, right?
Yeah, just put this on.
Oh, GMOs. Probably the one that we know in the most detail is recombinant DNA techniques. And you'll really want to remember that I think we had 5 different players that were important, right? So what does that mean? Recombinant DNA technology just means that I'm recombining DNA from 2 different species, right? So I am combining DNA from 2 species. And the example we gave was E. coli and humans, right? And it was to make an insulin that I called Humulin. It was the very first biotech drug. And we call this recombinant insulin because it wasn't made in human cells, it was made in bacterial cells. To do this, we need a couple of things. 1, I need E. coli to be my host. 2, I need the insulin gene. 3, I need plasmids. 4, I need restriction enzymes. And then the last one I needed was reverse transcriptase. Okay, coli is because proteins are too complicated for me to just put in a test tube and mix chemicals and think I can make it. So my host transcribes and translates for me. And of course, we know if I'm transcribing and translating, I gotta give a gene, I gotta give him the DNA, so that he can make mRNA, read the code, and make a protein for me. So I have this host, and the way that I get my gene into him is to put the gene into a plasmid. A plasmid is just a little piece of DNA, smaller than a chromosome. It looks big here, but it's really tiny compared to the E. coli. And the plasmid goes into there. Don't worry about the word transformation. In order to get my gene in the plasmid, I need the fourth one. And this is a restriction enzyme. So I take a plasmid out of E. coli, right? Take it out. I cut it open with, so it's still the plasmid, but now it's cut. And the thing that the molecular scissors, if I can draw scissors like that, they're called restriction enzymes. Yes.
So the plasmid is like the gene that's coming from E. coli.
It does have genes on it, but it's just a piece of DNA, and we call it a vector or a vehicle. It's just driving the DNA, because I can't really take a regular piece of my DNA and think it's going to get taken into E. coli. The plasmid does that for me. So I take the plasmid out, I cut him open. Remember, this guy here, he came from E. coli. He's part of the immune system of E. coli. And when he cuts DNA, his substrate is very specific sequences of DNA. So there are 1,000 different restriction enzymes, and each one will only cut a particular sequence. Like maybe it has to be GGCTTA. And if it's anything off of that, it's not going to cut it. Yes. And so I take the plasmid out, I cut it, I get my gene to go inside it, and then I put it back in, and now I have the gene that's going to be converted into that mRNA, which is going to be converted into a protein called insulin. And the last thing that I needed comes into play because I get it from viruses, so I talked about it in virology, and that's reverse transcriptase. Because you know that my gene, my human gene, has introns. And you also know that E. coli has never seen an intron. He doesn't know how to cut out introns like I do. So I take an extra step. Instead of taking my gene from the DNA, because that's a little bit of a lie, I actually go in and I take the mRNA, I convert it into a gene, and then I put it into the plasmid. Okay, so if I do that over here, mRNA into DNA, that's where reverse transcriptase comes into play. And you know that part of the processing of your messenger RNA is to remove introns. So if this guy doesn't have introns, then he has no introns. And always when you see the cDNA, you know that it was reverse transcriptase that made it. That's like copied or complementary. So you can assume when you see cDNA that it means there's no introns. Now E. coli can handle it. Questions on that part?
You said restriction enzyme cuts out the gene out of the water, like it cuts the water gene out?
Yes, yes. So what it'll do is two different things we add. We have the plasmid, right? And then we also have, first I explained to you, I'm saying it's going to cut a gene out of my chromosome. But really what it's going to do is it's going to cut my mRNA And I don't go into too much detail in this, in the exam, but, so here would be my gene, but I have overlapping sequences here where the restriction enzyme cut, and these sticky ends are gonna try to find the sticky ends that are in this. I don't go into detail on that, but both of them are cut, so that now this can go into that. Now, another difference between E. coli and humans is that I can do post-translational modifications. I add sugars to my proteins. But where did I tell you that was done? Golgi apparatus. E. coli, no Golgi. So we can't really do that quite. We can transcribe and translate, but that's about where it ends. Okay, so that's recombinant DNA technology. The other ones are really a matter of just knowing the definition of them. And so, for instance, the Human Genome Project was just sequencing all the A, T, C's, and G's in all the chromosomes, right? So here, sequencing all the 3 billion letters, A, T, C, D, in all of my chromosomes. Now, the important part to know about it is what we could do once we figured it out, right? We knew that we had 3 billion letters. We know we have 20,000 different genes. We already knew we have 46 chromosomes. We also found out that we're 99.9% the same. That's all humans. But we also know we have a lot of similarities. We call them homologies to other organisms. So remember we talked about that roundworm and the fruit fly. We have greater than 50% similarities to them. Now, when you look at that chapter in your book, they talk about functional genomes, they talk about ESTs, they talk about a lot of details that you're not going to in class, so don't worry about that. Questions on Human Genome Project? Now, gene therapy, in this therapy, unlike insulin, remember that I'm giving a protein, so it's a protein therapy, to the patient every day, multiple times a day. In gene therapy, I'm going to use a virus to bring in the gene that was broken, or the good gene, right, to the patient. Just like the plasmids, the vehicle, the recombinant DNA, the virus is the vehicle, or we call it a vector. And the one real life example that we talked about is there's a company called Spark Therapeutics, and they have the first example of gene therapies that are approved, and it's for blindness. Now, we learned about mutations, right? And we know that not every phenotype is dictated by one gene, but in this case, it is a blindness that's caused by one protein that's broken, or I'm sorry, one gene that's broken. So all we have to do is bring in that good gene for them, and they're cured. CAR T. Remember the T part is a T cell, so that's using your immune cells. You're going to genetically engineer them. And what does that mean? That means I'm going to give him a new gene. Give the T cell a new gene. And it's going to help find and kill cancer. is this. Pardon. I mean, although this kind of has, the same concept, all of these techniques for treating cancer are to target the drug to the cancer cell, so that we don't have all these negative side effects from being on treatments like chemotherapy. A magic bullet approach is when I take my chemotherapy, right, but I add to it an antibody. And that antibody, well, I have it the other way around, all right, sorry, like this. He recognizes cancer, right? There's a lot of different ways we can do this, but these are called, what is the acronym they use? They're antibody conjugated drugs. There's an acronym for it, I forget, but they put the antibody that recognizes cancer on the old-fashioned drug. Now that just comes here. That's called a magic bullet approach. We learned this antibody.
And you said gene therapy with the minuses, what was that?
That this gene therapy is when you bring the good gene as the drug. The drug is the gene, and Spark Therapeutics use that gene therapy to cure blindness. Now, number 5 is CRISPR, and CRISPR goes one step beyond gene therapy. Gene therapy can bring in a good gene if that's what you're missing. CRISPR can cut out a bad gene and bring you a good version of the gene if that's what the problem is, right? So, you know, when we talked about diseases like Huntington's, there's some bad stuff there. I need to get it out. It's not good enough for me to just bring in a good gene. So CRISPR removes a bad gene, brings in a good gene. No approved therapies. It shouldn't even be in humans yet, or maybe it is. And then the last one that we talked about here are just genetically modified plants and animals. And the only thing you really need to know here is that the vehicle or vector, Agrobacterium, it's called Tunafasciens, that brings the new gene to the plant. Again, this is a form of recombinant DNA technology because we're giving maybe a bacterial gene to corn, right? We talked about that, BT corn. So that's different species, recombinant DNA technology, but it creates a genetically modified organism. In biotech, were there other things I listed under biotech?
There's only 4, it's just about gel, electro.
Oh, gel electrophysical forensics, right, yes, yes. Okay. So, in all of these examples, well, this one, we're manipulating things, right? We're manipulating the bacteria, we're manipulating the patient's DNA. With the Human Genome Project, all I'm doing is looking at the sequences. And for forensics, I'm kind of doing the same thing. I'm just looking at your DNA and I'm comparing it to DNA that I found somewhere else. I might be solving a crime. I might be comparing it to some other person I want to know if you're related to. Maybe we're comparing it to samples that were left at like the World Trade Center. So in that case, we use PCR and electrophoresis. And PCR is just copying DNA. So whenever I have a little bit of DNA and I want to make a whole bunch, I do PCR. I then take that whole bunch of DNA, I add it to a gel. And remember what the gel does is it separates the DNA out by size. So this separates DNA based on size of the particles. Now that's kind of hard to see sometimes because it looks like, well, the band's the same, but that's not really what's in there. So here I might have a piece of DNA that's that big, whereas down here I have a little piece of DNA. And where that comes into play is these areas called STRs. If if I'm 99.9% the same as all of you, I need to look at a different region of the DNA to find our differences. And that is STR. So it stands for short tandem repeat. And what it will be is, let's say I have a repeat, I'm just gonna say GATA, and maybe I have one repeat, one that I got from my dad. My other chromosome has this, GATA, GATA. My dad's chromosome gave me 2 repeats. And when I run these on a gel, this might be 2 repeats and this might be 1 repeat. And then when I compare it to an unknown sample, the same number of repeats are kind of counted by electrophoresis, and I compare my banding pattern. So maybe there's one that's even higher up here that's 3 repeats. Something, oh yeah, down here that's lower. Remember, these STRs come from non-coding regions, and what they are is just places where there are no genes. So if I look at my chromosome, I have gene 1, gene 2, gene 3, but all of these regions here are non-coding. So that's really where I'm going to look at these repetitive areas, and running the gel allows me to see how many repeats I have and compare my repeats to somebody else. Yeah.
What was the 13 loci?
Oh, okay. I don't just look at one place, like if this is an STR short chain and repeat, I might look at, this is one short chain and repeat area. This is another one. This is a third one. And when I look at all of my chromosomes, these tests are really done on 13 different places to make sure that I'm really able to discriminate you from somebody else.
Okay, thank you.
Yes.
What's the difference between adult and stem cells?
The difference between,
Adult cells and stem cells?
okay, we didn't really talk about that in class too much, so I did take the question down about embryonic stem cells. But what I do want you to know, and you guys saw this question, you'll see it like verbatim again, is that if I take a fat stem cell out of myself and I put it into a petri dish and I give it nerve growth factor, it develops into a nerve cell. It's got that flexibility. That's all about it turning on different genes, signal transduction. And so all adult stem cells have that flexibility. Embryonic stem cells, just to let you know, they come from a fertilized embryo, right, that's 5 days old. They were very, and remain very controversial because you're destroying the embryo, what would be a baby. But these aren't ever embryos that were going to be implanted into a mother anyway. But that's a different thing. Just know that adult stem cells, you have 200 different kinds of them in your body. Once I take them out of the body, I can manipulate them with these factors to get them to be retrained and reprogrammed. Other parts in that chapter that, let's talk about signal transduction again. This comes under stem cell technology, it comes under cell communicate, it comes under cancer. And this idea of signal transduction is that you have a cell, we're a multicellular organism, and this cell has DNA in its nucleus, and the outside of the cell is filled with messages that can't always get inside the cell. And there will be a protein, a receptor on the cell, where the message docks. Okay, so this message is usually a protein, right? We might call it a hormone, sometimes it's called a growth factor or something like that. They're really all the same thing.
Oh, so this is our signal transduction
This is-
process of.
Yeah, this is the general signal transduction pathway. And then I'm gonna talk about P53 when we get to that part. Signal transduction. guys touch each other, they change shapes. And so now this touched him, he changes shape. And then there are proteins inside here. When this guy touches this one, he changes shape, he changes shape, he changes shape. These we call relay proteins. And sometimes you'll notice that they're kinases. But the message just goes from here to here to here to here. We're whispering down the lane. And then eventually the message goes from number 5 to a transcription factor. And the transcription factor knows how to find the gene that needs to be turned on. So this gene matches the message up here. And that transcription factor will sit right down here on the DNA. And remember, that calls in, it sits down on a switch. And it calls in RNA polymerase. And then we go through transcription of gene. Okay, so that's how it normally happens. I'm gonna stay on this one to talk about P53, but then I'm gonna show you like other hormones that actually can get in the cell and what they do. Yeah.
You said it causes RNA polymerase to come into transcription?
And start transcription. Yes. So if you look, it's this, it's transcription factor, which is another protein, on a switch. The transcription factor calls RNA polymerase to the scene. And then RNA polymerase begins transcription. Yeah.
Whatever is how,
Right, exactly. So this message could be saying grow, divide, mitosis, or the message could be saying stop growing. And depending on this message, you're going to turn on different genes inside. So my stem cells, they have to divide all the time. So the signal they're getting from the outside is grow. Laura needs more intestinal lining. But the other cells in my body are getting a signal to say stop growing because we don't like cancer. Now, the P53 and where this comes in cancer, this transcription factor here, there's lots of them, but for example, P53 is a transcription factor. And in the case of P53, he sits on the switch, he calls in RNA polymerase. RNA polymerase starts transcription. So for P53, he starts transcription of a break, like a break in your car. Whatever that protein is, it's going to stop the cell cycle. So he's a good gene. But 50% of cancers ensue because that gene is broken. And if P53 is broken, right, so now let's say P53 broken, 50% of cancers, it can't sit on the DNA, it can't sit on the switch like it's supposed to. If it can't sit there, it can't call in RNA polymerase. Polymerase does not know transcription. And then the cell, no break. So the cell grows out of control and leads to cancer. Again, P53 is just one example of how the signal transduction pathway happens. It's just one example of a transcription factor. Yes?
Are we supposed to know any more transcription?
Yeah, that's good. The only other thing that I want you to know about signal transduction, has to do with, remember we talked about testosterone? And in testosterone, we still have the same thing where we have a gene that we want to turn on, and we still have a receptor, but the receptors are inside the cell. So these are all receptors. Testosterone, because it's lipid-based, right, it can go right through that membrane, and it finds different receptors. Different receptors, I'm going to expand this chromosome. Testosterone with this receptor might sit here and act as a transcription factor. Testosterone with this receptor might sit here, and this one sits here. 3 different genes can be turned on by one hormone, but multiple receptors. So the testosterone plus receptor, that's a transcription factor. Transcription factors are always turning genes on. And the reason that that's important is there'll be a question on there that says, why do you have so many different phenotypes based on people taking hormones, anabolic steroids? But the same thing can happen with estrogen as well. So these get in, they sit down on different receptors, you sit down on different genes. So you get roid rage, you get acne, you get muscle buildup, you get loss of libido. All these things are happening because testosterone has a lot of partners inside the cell.
Okay, never mind.
Okay. All right, so we're all good with this, right? How does AZT inhibit HIV? Remember, HIV is a retrovirus. Retroviruses are made up of RNA. And they get inside your cell and they go backwards and make DNA. So here is the virus's genome, right? He is RNA. He gets inside my cell, he becomes DNA. And what's that protein that does that? Reverse transcriptase, right? And if you think about enzymes like you learned them in chapter 8, right? We always show them like this. The substrate is going to get in here, and AZT binds where the substrate should be. And when you do that, that's active inhibition, or a competitive inhibition in the active site. So inhibition in the active site. So I call that competitive. Don't worry about the analog, but the T thing is like finding, it looks like finding. I don't go into whether the 3' hydroxyl, even when I talk about RNA replication, I don't ask you about that. Yes. Oh, the homologous structures. So homologous structures are similar, right? Just like you have homologous chromosomes, right? So similar structures in evolution could be that my arm looks like the leg of a horse. It has a similar bones and organization. That's structural. So think about the adults versions, that's structural homology. Developmental homology is when I take a look at the fetal development of different organisms and I see that they look similar early on. We look really similar the further back towards fertilization we are, and then as we develop in utero, we look unique. Then the genetic one is I'm going to look at DNA. I'm going to look at the sequences in the genome, and the big example we gave here, remember, was that based on structural homology, I put whales off on a different branch away from hippos. But once I was able to sequence DNA, I now know that hippos and whales are more closely related than other land-dwelling animals. Yeah, it's because, when Darwin put them together, and Linnaeus and everybody else who did it, they only could see the physical things, they couldn't see inside the DNA. And so now we're learning that just because you don't have an ankle bone doesn't mean that you weren't related, that you are close cousins.
This is homologous structure.
These are homologous, yes. Yes.
What's for evolution, what's left in pressures for golf for the evolution of skin color?
The what?
For the evolution of skin color.
Oh, yes.
When the sun is down on us,
Yes, yes, so in that, like, I kind of went in kind of a long drawn out, you're gonna wanna know that vitamin B and vitamin D are the key factors that drove evolution of color. That UV light destroys vitamin B, which is needed for reproductive purposes. Melanin comes in to save the day, it protects the vitamin B. And then as you move away from UV light, you need that sunlight, but now you're not getting a lot of it because you're living in higher altitudes, or higher longitudes. And, so you don't get enough vitamin D, which is also needed for my babies to be healthy. So vitamin B and D are the driving forces in skin color.
Vitamin D. Didn't you say A?
Yeah, the melanin protects vitamin B. I'm sorry, did you have another question over here? Oh, okay. Yeah, it protects B.
Just out of curiosity, is the genetic or the structural part more related to taxonomy?
Taxonomy would be more structural. And again, that's because when we first put taxonomy together, we didn't know all the genetic stuff yet. But I think what we'll see is, we're gonna rearrange things, just like we are with the whale and the hippo. We'll start rearranging that taxonomy once we understand the DNA better.
Yeah, okay, thanks.